Good afternoon, good morning. I'm Hisham Ben Youssef, professor at Mohammed VI Polytechnic University. I'm also the manager of the High Throughput Multidisciplinary Research Laboratory. It's my great pleasure to be the chairman of this last session and second workshop entitled Sustainable Development of Nanotechnology in Agriculture. Let's start, let's say, of our first speaker of the session. Then I am having really the great pleasure to meet him now and let's say to see him for the first time and also to present him as our first speaker. François Perrault is an assistant professor in the School of Sustainable Engineering and the Built Environment at Arizona State University. He completed his PhD in environmental science at the University of Quebec in Montreal and was in his ERC postdoctoral fellowship in the Department of Chemical and Environmental Engineering at Yale University. His also research was recognized by the Emerging Investigators Award of the Sustainable Nanotechnology Organization and Quentin Mees Research Award from the Arizona Water Association. He is explores the sustainable use of nanotechnology for environmental applications with focus on bio-nano interactions, biofouling controls, and also membrane-based water treatments. Professor Perrault, the stage is yours.
Thank you. Let me just put myself on full screen. And there you go. Let me know if you don't see well my presentation. Hi, everyone. It's a great pleasure to be here virtually. First, thank you to the organizer of the conference, to the chairman, Dr. Ben Youssef. And it's a great pleasure to kind of kickstart this workshop. So as part of the workshop, I was asked to give kind of a brief introduction on nanotechnology and how we can basically use the unique properties of unique opportunities of nanotechnology to basically enhance plant production, how to use it in agriculture. So for those of you that are familiar with nanotechnology, this might be a little bit of a review, but for those of you that are not familiar, I hope it will help you understand a little bit where we're going and why we are interested in the things that we're doing. So there's some formatting issue there, but that's okay. So yeah, just a brief introduction, and thank you, Dr. Ben Youssef, for the very kind introduction. Yeah, my name is François Perrault. I'm an assistant professor at Arizona State University, and most of my work really focused on biological interactions of nanomaterials. And we've been looking at these kind of interactions with all sorts of biological systems, and actually starting with plants. The first model that we used were aquatic plants, looking at the potential environmental impact of things like metal oxide nanomaterials or cold nanoparticles in aquatic plants, looking at change in photosynthetic activity and growth, pigment production. And then over the years, we moved to work with microalgae, with bacteria, biofilms, in vitro cell culture, kidney cells, neuronal cells, and now we're kind of exploring actually 3D cardiac cell tissues. So we're really all across the board in terms of the type of biological system. And fundamentally, really what we're interested in, the main question that we try to address is how can we tailor the properties of nanomaterials to guide what kind of interactions we're going to see with biological system. So plants, of course, for the first model, there always a third model to work with. So I'm very glad to be exploring these opportunities for agriculture. And today we're going to discuss specifically on these interactions with plants and what makes them special as nanomaterial. So the presentation will be divided into kind of different components that you can see here. First, I'll talk about nanomaterials, nanotechnology, give a brief, really broad overview of what are nanomaterials and nanotechnology, and then focus on the type of biological interactions that were interesting when it comes to nanofertilizer and application in agriculture. And then I'll connect basically these type of biological interaction that really started kind of in the biomedical field with what we can do in agricultural applications. I'll talk briefly about the different challenges and barriers to address, and then I'll finish with some concluding remarks. So first, well, what is nanotechnology? And you might have heard of nanotechnology in some ways, because it's been a very hot topic of research for the past couple of decades. But very often nanotechnology are kind of defined as anything that has one dimension below 100 nanometer, between 1 and 100 nanometer. So in that definition, nanotechnology can be anything from organic, inorganic, biologicals, polymers, really anything can be a nanomaterial. I think a more elegant definition really comes from this definition from the NNI, the National Nanotechnology Initiative, that said that the nanotechnology is the understanding and control of matter and dimension of roughly 1 to 100 nanometer, so we have the same scale size, where unique phenomena enable novel application. And the key word here is really this unique phenomena. There's something different about materials when they enter this 1 to 100 nanometer that makes them different from bulk material. And nanotechnology is really about exploring and exploiting these unique phenomena to try to enable novel applications. So example of these type of applications that we can be looking at, two very classical kind of properties that we've seen in nanotechnology. The first one being optical property. This is what you have on the left. You can see basically the fluorescence emission spectra for different types of quantum dots that are very small inorganic nanoparticles. And you can see that very small change in size in these nanomaterials, less than 1 nanometer, 0.5 nanometer, can lead to very significant shift in the optical property of your particle. And so material scientists in nanotechnology seek to exploit these small changes in size to tailor the optical property, the physical-chemical property of the material to really match exactly what is needed for the application. On the right, you have another very classical example in terms of surface reactivity or catalytic property. And then you see the effect of size on the reactivity per unit of particle. And you can see that there's a clear increase in reactivity as you decrease the particle size. And if you recall that 1 to 100 nanometer threshold that we were discussing a bit earlier, well, this is your kind of 1 to 100 nanometer here that you have on this graph. And you can see that when you go below that 100 nanometer range, surface reactivity and surface property really starts to pick up very quickly, because below that range you start to get a much higher proportion of atoms at the surface of the particle that becomes available for surface interaction. So nanotechnology is really about controlling these surface properties that becomes dominant compared to bulk properties when you get to these very, very small properties. So, so that 1 to 100 nanometer size range is not completely arbitrary, but what's more important is the unique property that emerge at this size range. And these are really just two very simple property example, but there's really a lot of different properties and applications that has been explored for nanotechnology. And it really stems from the very broad definition of what is a nanomaterial. As we can mention, it can be inorganic, organic, biological. So that makes it a very broad set of property that can be utilized for commercial product. So here, this is a very interesting survey that adapted from the work from NanoWork, where they looked at all the commercial product that claim to have nanomaterials in their composition, and what are the properties that are being leveraged from these nanomaterials to enhance the performance of the commercial product. And you can see that they're very broad. I tried to group them in terms of color when we have kind of mechanical property, biological property, kind of corrosion resistance. But you can see that we have property based on the type of material that can be included. And you have things like antimicrobial applications where we can embed silver into fabrics to make odor resistant clothing or socks or any sort of basically fabric that you want. Something that is very interesting for the biomedical setting. You can have super hydrophobic coatings using nanostructure surface property to have a waterproof coating. So this is something that you have maybe on your car window. You can have self-cleaning surfaces. And this is a nice church and round that is all white and stays white because it uses these photocatalytic properties of titanium dioxide that basically take UV light from the sun and use it to degrade and self-clean itself just using sunlight. And then you can have very high strength material like polymer that are embedded with carbon nanofiber to provide them with very high mechanical properties. So wide range of property, wide range of potential application that kind of reflects basically the diversity of materials that is integrated into what is a nanomaterial. So when it comes to the biological interactions of nanomaterials, which is really what we're going to focus about, there is the same idea that you can tailor the property or how the cell will respond to the exposure to nanomaterials by changing the size of your material. And this can be done basically just by changing the size or the surface property, the surface chemistry. So here, well, one thing to keep in mind is that cells are naturally equipped to deal with particles in the sense that a lot of the cells in our body will basically take up particles when they encounter them. But the mechanism by which they will take it up will be dependent on the size of the particle. And by changing the size, you can change the way that that particle will be taken up by the body. So if you have bigger particle, you might do something like phagocytosis. But if you have much smaller particles, there's other types of mechanisms that are involved. And then, of course, there's a lot of unknowns. There's a lot of diffusion or unspecific mechanisms that have been proposed, and there's still a lot of research on that. But cells naturally take up particles, and that's a way basically that we can use particles to deliver things into the cell. Something that became interesting very quickly is the fact that nanoparticles actually can bypass some of these biological uptake mechanism and slip cell security, if we can say, where simply by interacting with cell membranes, they can essentially penetrate passively into the cell. And this becomes very interesting because there's a lot of biological barriers that can be hard to cross. So engineering a nanocarrier that would be able to slip cell security became very interesting for things like delivery of biologically active chemicals into the cell. So the first field that took advantage of this opportunity, this property of nanomaterials to slip cell security, of course, is the biomedical field. This has very important applications when it comes to drug delivery or antibiotic delivery, because then you can reduce the toxic load and reduce the amount of chemicals entering your body. If you can see here on the left, there's kind of two main examples of how you can achieve this. You have in the little teal blue particles, here it's a liposome, but it is used for passive targeting in the sense that this particle will be able to cross cellular barriers and basically enter the body and release passively in a controlled way their load, their drugs, or their anti-cancer drug or antibiotic. Alternatively, what you can do is have very specific binding sites, and these are the yellow particles where you can basically achieve very specific binding of the particle to a specific cell, let's say a cancer cell or a bacteria, to achieve very specific delivery of your chemical into the cell. And in some example, actually some very interesting example where applications where magnetic nanoparticles like zero-valent iron would be used, and then you can use something like a magnetic field to guide the particles to exactly where you want. So here you have this little mouse that unfortunately had cancer, and then you could use that magnet to basically force the accumulation of particle into the cancerous region of the mouse. And all of this basically is really to try to take our bioactive compound and keep it into what is called a therapeutic area or therapeutic zone, and avoid basically overloading the body with a toxic level of chemicals. So of course, this has tremendous applications in the biomedical field. That was something that was adopted really early on. And you can see kind of a timeline. It's a slightly outdated timeline. This paper is 10 years old, but it really kind of represent the main milestone that I would kind of like to emphasize in this presentation. There's been a lot of innovation, but some of the kind of important one would be things like control release polymers. And this is kind of the first step really in that use of nanomaterials, nanocarriers for drug delivery, is to have a control release platform that can just slow down the release of your chemicals. And then as things progress, people have looked into things like targeted delivery, where you can have specific ligand on the particles that would dictate where that particle will go into the body. So now it's not just control release, but you're really trying to deliver something specifically. And then as things becomes more complicated, the thing disappeared. But as things becomes a bit more complicated, then you can have a co-delivery of different compounds that can act together to basically allow for a synergistic interactions between the different components. So why is that important for agriculture? Well, if you think of what is the main challenge of using nanomaterials in agriculture, well, the main challenge is chemical use efficiency. When you apply these fertilizer to field, of course, you have very, very low efficiency, more than up to 50 to up to 80, 90% of what you apply to the field will simply be lost. So if we can engineer nanocarriers to be able to increase this chemical use efficiency, then we'll have a tremendous improvement of the process. And if we look only on phosphorus, because we're in a conference about phosphorus, right? There's kind of two main competing pathways that kind of limit the efficiency of phosphorus to the plant. The first one, of course, is leaching. You're kind of losing your phosphorus into the soil. But the second one is competition with microorganisms. And I like these two because these are the two kind of mechanisms that there are opportunities for nanotechnology to address these kind of limitations. So I'll present kind of three examples of how we can achieve chemical delivery in agriculture using nanotechnology. First, by looking at controlled release of nutrients. So just slowing down the release, the controlled delivery of nutrients, and then how we can bypass cellular pathways, basically exploiting the unique biological interactions of nanomaterials to try to do things to deliver chemicals in a very different way. So the first one, if we go back to our analogy with the biomedical field, if we think of the controlled release of chemicals into the body, similarly in agriculture, the first step was to develop controlled release fertilizer, where the goal is to simply extend the lifetime of that fertilizer. And this has been done by using basically inorganic nanostructures, by coating polymers, coating fertilizer granules with kind of a polymer coating, by having biomaterials that you can basically kind of infuse with nutrients, and then they kind of slowly release their loading over time. All these strategy, basically, the goal is to extend the lifetime. And you can see here an example on the left. This is a urea-based urea hydroxyapatite fertilizer, and by engineering a nanostructure in that urea-based fertilizer, they were able to extend the lifetime of release by more than order of magnitude. So you have a much lower release of urea. And then the more important information is that when they applied this to a field to evaluate, well, is this nano fertilizer really better? They saw an improvement in crop yield for the nano fertilizer compared to the granular commercial fertilizer. But they saw this, and this is something that is not indicated on the graph, but they saw this at essentially 50% of the dose of the granular fertilizer. So half of the fertilizer was used, but they achieved higher performance, which really show the potential, the capacity of slow release compound in enhancing plant growth on the field. So there's really a great opportunity to kind of reduce the release of compounds. And remember that these fertilizer that are leaching out into the field will end up into the environment, creating eutrophication, ending up into our wastewater supplies that maybe have to pay a lot of money to remove. So improving the efficiency really has more than just benefit in terms of chemical use. So here on the bottom right, there's a very important study that was published recently in Nature Nanotechnology, where they look at essentially a meta-analysis of all the controlled release devices that were done for agricultural fertilizer. And what they found is that when you look at the nano to non-nano ratio, which basically a value more than 1 indicate an improvement compared to the non-nano form. You can see that all different types of nanofertilizer perform better than the non-nano form for all types of macronutrients. But most specifically, when you look at macronutrients fertilizer, this is really where all the opportunity lies, and the most promising opportunities can be achieved. Then moving forward, again, taking back into our analogy with biomedical delivery, we can go from control release to control delivery. And this is something that we've been exploring in my lab in the past. We're trying to basically take inspiration from nature and try to look into these very specific interactions that you can have at the root surface using lectin binding sites, for example, and then engineer our nanocarriers to have very specific receptors. Here, this is a modified form of glucomannan, a carbohydrate that will specifically bind to that lectin to really enhance and enrich the root surface with these nanocarriers in order to be able to achieve controlled delivery of that compound. Another similar approach to achieve basically a smarter, more controlled delivery of that compound, not just a release, but trying really to target our delivery of nutrients really where it matters, is to use kind of a smarter material design that could use basically a stimuli responsive polymer design. So this is a very interesting, very smart approach to do this controlled release, where they use basically a polymer that is pH sensitive, and then since the roots with these exudates change the local pH environment at the root interface, when the particle gets in the close vicinity of the root, the change in pH will trigger the release of fertilizer. But when the particle move away from the root, well, the particle then basically closes down and prevent the release from the fertilizer. So this is a way to really tailor where are you delivering your fertilizer. And in a sense, this allows you to try to mitigate that competition with microorganisms that I discussed a bit earlier by really targeting your release where it matters, at the root surface. And there's a lot of opportunities for this platform of stimuli responsive polymers. This is a study of kind of just stimuli responsive and polymers in general, and then specifically at agricultural formulation, looking at what is being done in terms of stimuli responsive polymers. And the main ones, of course, is pH, but things like temperature or light, production of enzymes by the plants are all things that you can explore to try to really control the release of your chemical fertilizer to where it matters for the plants. And then the last example that I want to give is about how we can exploit these unique biological interactions of the particle to enhance the uptake and efficiency of the chemical by the plant. And this is a very interesting development that has emerged in the last couple of years, where people have looked into foliar applications of these nanofertilizer. So instead of applying to the soil, you can apply it to the leaf. And what happens when you apply it to the leaf is there's very interesting size-specific uptake mechanism that can happen. Very, very, very small particles are able essentially to travel into the cracks of the wax layer on the leaf surface. But as you get bigger particles, of course, they're not able to cross that wax layer as easily. But what you can do is enter through the stomatal entry, and there particles up to about 10 nanometers are able to enter easily into the plant. So that's a way to kind of avoid all the soil interactions, avoid the leaching and the erosion and the competition with microorganisms, because now you're feeding your chemical directly into the plant. And then the main question when you apply foliar delivery is, well, does it stay into the leaf? You don't want that. But nanotechnology have found that you can control where the particle goes into the plant by simply changing the size, the shape, the surface chemistry of your particle. For example, if you use a cubic particle, you can enrich the root system compared to the leaf. And this is all for foliar application. It's just that the particle relocate from the leaf to the roots, and then changing the morphology of the particle can really guide where it is going. Similarly, you can do the same thing. You change the surface chemistry or the surface size. I won't go into the detail of this graph that you have here on the right, but you can see that particles of 3, 10, 50 nanometers. If you change the surface chemistry from citrate to PVP, you can then change where it is going into the plants. So there's really an opportunity to guide where your particle is going. So there's really a myriad of opportunities for nanotechnology. We're really focused on kind of nano-fertilizer, but you can do the same thing with nano-pesticides, nano-herbicides. There will be a very interesting talk about nano-sensors, how to use these nanoscale property to detect things into the plants, to monitor cell viability. So there's really a lot of different things that can be done with nanotechnology, and that just reflects essentially the wide diversity of nanomaterials that can be engineered from material science. So moving forward at our main challenge, well, this is where the analogy with biomedical applications kind of break down, because scalability and cost will be extremely important. People are willing to pay for nanomaterial that can cure disease, but when you think of how much nanomaterials can be applied as nanofertilizer, scalability and cost would be extremely important. Environmental footprint, again, will be also very important, simply because you don't want to create a problem and trying to solve another one. And for this, we really need a big picture kind of life cycle point of view. And we'll have a presenter today that will also discuss about this aspect of trying to do it in a sustainable way. And then the last challenge is really about social acceptance. There's been a lot of pushback on nanotechnology that is not really that different from the movement from GMOs. So we'll need very careful science communication to make sure that we communicate that our technology is safe and we do all the proper risk assessment. So as concluding remarks, nanotechnology really offers opportunity to fine-tune the properties of materials to the need of the application. And then the nanotechnologists can really exploit the unique biological interaction of nanomaterials to improve chemical delivery. If you avoid the soil interactions to leaf application, you can really reduce fertilizer loss, and then the economical and social barriers can be addressed. I want to thank you all for your attention, and I'm really looking forward to this beautiful symposium that we have for today.
Thank you very much for this nice presentation. It was very, very interesting. I think the session of questions is started. I don't know if there is a question from the audience. If not, I can start with the first one. Yeah, I see. Okay. From Thank you, Francois, for this nice presentation. I'm wondering if nanotechnology or nanobiology will help agriculture to face, for example, symbiotic stress or even biotic stress.
Yeah, this is a broad question. I would say yes, in the same light of what I discussed, in the sense that you can, instead of using fertilizer, you can load nanopesticides. People have looked into using copper as a fungicide to mitigate, essentially, to prevent pathogen growth. So in the same area, you can control the amount of chemicals that you use and really try to minimize leaching into the environment. So these are toxic chemicals, of course, that you use. So if you can minimize how much is being lost, how much is wasted, then you have a more sustainable process and similar application.
Okay, I see there are other questions from Haula Ukaltuma. Thank you for this interesting information. Would we apply nanophosphorus fertilizer through foliar application?
That's a very good question. To my knowledge, this has not been done yet. Most of the research I've seen is more about micronutrients applications, things like copper. But I don't see why you would not be able to do it, because the interactions is really guided by size, shape, surface chemistry. So if you can achieve the same size, surface chemistry for a nano phosphate fertilizer, then you could have the same kind of fate and transport into the plants.
Okay, I think there is another question from Ilham Mardad. Thank you. We can say that the nanotechnology is the future of agriculture. How about the cost of this technology, especially in Africa, where the need for more yield and nutrients in food is growing?
So yeah, this is what I mentioned in my last slide. Scalability and cost will be the main challenges. There have been a lot of development of trying to do these control delivery, control release platforms with very sustainable material, things like chitosan or kind of biomaterial. So very cheap material, very sourcing material. So now we just need essentially to find a production process that can scale it up to large scale. Maybe fertilizer is something that would be hard to compete with, especially when you think about things like pesticides, where things become a bit more expensive and then the impact are toxicity. Then there might be a good platform to start. But I really believe there's a good opportunity for nanotechnology to improve chemical use in agriculture. And when you think of the scale of the problem, just a few percent in improvement in chemical use would have huge impact at the global scale.
Okay, thank you. I think we have another one from Rahim bin Zawiya. How we can use nanotechnology to optimize water consumption?
So I'm assuming this meant water consumption by the plant. That's a good question. Actually, that's not something that I'm too familiar. If I would make kind of an hypothesis, you can probably have some sort of kind of hydrogel that could retain water locally near basically the plant, and then basically you can store water in when needed. So that I think would be my approach, but I'm less familiar about this.
Okay, I think we have maybe a last question from Hisham Fniri. How can one address crop versus the weed selectivity in a field using nanotechnology?
That is a very good question. I guess there we will have to rely on a combination of strategy, and that could use some of the current strategy to try to select crop versus weed, and just combine it into enhancing plant production for the field, but then use another alternative strategy. That's not something I thought about. There might be a strategy, but it's not coming to my mind.
Okay.
Good question.
I don't know if you want to add something on it, but if it is not the case, I would like to thank you again, Professor Pero, for the question, for the answers, and I think we can move to the next talk. Thank you very much. I think our second speaker of the session is Professor Liane Gilbertson. Professor Liane Gilbertson is an assistant professor in the Department of Civil and Environmental Engineering at the University of Pittsburgh. She received her master and PhD degrees from Yale University in the Department of Chemical and Environmental Engineering, then followed by a postdoctoral associate fellowship in the Center for Green Chemistry and Green Engineering at Yale University. Her research group at the University of Pittsburgh is currently engaged in projects aimed at informing sustainable design of emerging materials and technologies proposed for use in areas at the nexus of the environment and public health. Dr. Gilbertson also research has been recognized and is supported by the National Science Foundation, 3M Non-Tenure Faculty Award, and the Ralph E. Poway Jr. Faculty Enhancement Award. Thank you very much for accepting to be our guest and to present this your work here, and the stage is yours.
Great, thank you. Can you hear me? Okay, my sound coming through? Hello? Can you hear me?
Yes.
Okay, good. Great. So thank you very much for the introduction, and it's really a pleasure to be a part of this conference, and I really appreciate Francois setting up the session here with a great introduction to nanotechnology. I'm going to shift the focus a little bit to talking more about when we design these applications, so a lot of the things he was talking about at the end there for delivery of macronutrients and pesticides to plants, how we should be doing that and thinking about it from a systems perspective. And so the reason that we're here in this session is because there's an incredible need for solutions to producing food for our growing population. And more, it's recognized that we need to do this sustainably, most notably recognized by the United Nations Sustainable Development Goal, specifically goal number 2, which has to do with food and particularly promoting sustainable agriculture. What that means and the opportunities across the food production system are really numerous. What I'm going to be talking about today is specifically related to crop production. And so to start thinking about this from the system perspective, I think it's really important to think about all of the things that are necessary to grow the 3 billion metric tons of crops that are produced globally each year. So first, this requires a huge amount of inputs. And so there's a lot of staggering statistics that I could have included here, but I've taken just a subset of those. And if we just look at the fertilizer and pesticide use, the water use and the energy use, these are really staggering numbers. And so if we think about producing more, we need to think about how can we maintain and ideally reduce the quantity of inputs that are needed for crop production. This is called sort of crop and agriculture intensification, growing more with less. Crop production also relies on natural resources, and particularly 2 finite resources. So first of all, land. and it's finite and actually depleting. If you look at the soil erosion that's occurring across the world, we're actually losing our cropland due to soil erosion, and that's primarily due to the overuse of all those inputs that I just talked about. We also have, of course, the conference is solely focused on phosphorus, and certainly others have likely talked about the actual peak phosphorus year and how much time we have left to deplete these resources, and perhaps they can be debated. But the idea here is that we don't have endless supply. And so at some point, we either have to be reusing what we already have in cycle or thinking about alternatives to this essential nutrient for crops. And then the final aspect that I want to touch on is the fact that all of these inputs, and particularly our inefficient use of them, has incredible impact on our environment. So specifically through emissions to the surrounding water bodies of nitrogen and phosphorus, we cause eutrophication. And in the US, this is a major problem for our coastal waters and also our big lakes and rivers. In addition, for example, nitrogen that is not used by the plants can be released as a greenhouse gas that is 300 times more potent than carbon dioxide. And so we have emissions that are contributing to global warming. And then, of course, there are pesticides and also nitrates that are released to water sources of drinking water. And I wasn't able to find a number of potential cancer cases that are caused by these emissions, but there's no doubt health effects that are associated with the excess that we apply to crops. And so, we're here in this session talking about nanotechnology and trying to develop technologies to use these inputs and prevent the impacts, use the inputs more efficiently, prevent the impacts on the environment and public health. And I just want to highlight the fact that we've done this in the past. So Francois had was able to do something there with his cursor. I cannot. But anyways, I have 3 examples of things that we've done in the past. For example, using crops for fuel, so corn-based bioethanol. And the idea here we had was to offset a non-renewable energy resource. But what we found after producing corn-based ethanol is that it's incredibly land-intensive, it's water-intensive, and it diverts a food crop. Chlorine has a similar story, although we still are using these things today, right? So we're disinfecting water, we're trying to get rid of bacteria and viruses, which cause a lot of health effects in drinking water. Yeah, chlorine's an acutely lethal substance, and so when we transport and store this chemical, it's a cause of concern. And then there's also more recently the realization of excess chlorine in drinking water causes disinfection byproducts. And then there's complex fluorescent light bulbs where we have energy efficiency increases, longer lifetime of the light bulbs, but it relies on the use of mercury, which is a known neurotoxin. And so now we have sort of this issue at the end of life of these bulbs where we've mobilized, again, a very hazardous substance in our sort of technosphere. And so what we want to do with crop production and the development of these technologies is ensure that these challenges that we are trying to attack with the best intentions, with exciting new technology, is that we design them responsibly to avoid these unintended consequences in the future. And the things that we might be concerned with, Francois touched on a few of them related to environmental and human health, but also embodied energy, which I'll be talking about today in particular, and then of course cost, economics, and ensuring that safety and consumption of the crops that we grow with their use. So the two questions that I'll be addressing in today's presentation in thinking about how do we approach technology development from the systems perspective is specifically looking at the efficiency of nitrogen and phosphorus-based fertilizers across the crop production system. And we did this to identify the greatest opportunity for improvement. Where could we intervene for the greatest gain? And then whether or not nanotechnology can be competitive over current alternatives, and ideally to offset a fertilizer use and the environmental impacts that are affiliated with it. I'm showing these 4 different publications here that have been featured on covers of the various journals, because this is where the majority of my talk lies, and so I don't have time to go into detail on everything that's included in these publications, and so I want to direct your attention to them for additional information. So the first question, what's the efficiency of nitrogen and phosphorus as we convert them from cradle, so from the isolation of phosphorus and nitrogen to the farm gate, so in the production of the crop? And so what we did in this particular study was look at 3 different stages, so synthesis, taking nitrogen from the atmosphere, the Haber-Bosch process to produce liquid ammonium, and then from liquid ammonium into the variety of nitrogen-based fertilizers that are then used on crops. Similar mining of phosphate rock, getting it into a form that we can now apply to crop fields. Then there's assimilation by the crop, so different crops will have different demands for nitrogen and phosphorus and incorporate that into their final crop. And then the nutrient content in that final product, and we use corn as an example. We did not, in this study, we're not accounting for the loss and the eutrophication. We're really just interested in what's called converting the atoms of phosphorus and nitrogen all the way to the incorporation of the product, and specifically looking at how efficient that process was. And in order to understand this new metric of atom conversion efficiency that we proposed in this paper is an understanding of atom economy. And atom economy is one of the 12 principles of green chemistry, specifically number 2. And so this defines the percent of total atoms in the reaction that end up in the product. And so you want a higher number here. You want more of what you put into the reaction to actually be incorporated into your desired product. And so it's a fairly straightforward calculation, as shown here, the mass of atoms in the desired product over mass of atoms in the reactant, and you multiply that by 100. What we went on to say is that if we can combine atom economy and other established metrics to get at an efficiency for the entire system, from synthesis to farm gate. And when we did that, so we have atom economy, we have nutrient use efficiency, so that's how efficiently the crop's using nitrogen and phosphorus. There's how much of the nitrogen and phosphorus that's taken up by the plant that's actually converted into the kernel. So that's the component that we actually can eat, or that animals can eat, rather than the stalk and the leaves and the cob. And we looked at 6 of the predominant most used fertilizers. And the conversion, if you look at the system, right, we always talk about nutrient use efficiency on farm, and we're saying that that's like, crazy low, and we need to be improving this number. These numbers perhaps are outdated and need to be updated. So these are the maximum nutrient use efficiencies. But if we look at the efficiency across the entire system, it's actually pretty embarrassingly low. So for every atom of nitrogen that we take out of the atmosphere, 8% of that, by the time you get to the end of the system, is actually in the food product. The same would be true here for, say, phosphorus that's taken from the ground, that's then used and taken up by the crop and then put into the corn kernel. We're at 5%. So these are really, really low efficiencies, and therefore there's a huge opportunity here for gains. But the question was really, where's the greatest potential for intervention? And if we look at synthesis, so this is just an example, and we did this for each of the fertilizers, but this is the example for urea. If you look at the chemical reactions that produce urea from liquid ammonium, these are really well established, and so we're constrained by the chemistries in which this occurs. And so a lot of work in this area has been around reducing the energy demand through catalysis. And the potential for alternative sources of nitrogen, I think, will be driven by cost, because there's endless amounts of nitrogen from the atmosphere, and so it's an unlimited supply. If we look at the incorporation into the final product, so that last stage, these are really constrained by the biochemistry of the crop. And so we could potentially alter this through, for example, genetically modifying the seeds or the crops to be able to incorporate more of the nutrients. And I think that there could be some potential opportunities there, and perhaps people are already working on that. The whole GMO movement, though, will have some societal challenges to overcome. And so we're not, that's not an area that we foresee as nanotechnology being the predominant solution. Where we do think nanotechnology can play a big role, and other technologies generally, is on farm and trying to improve this nutrient use efficiency. And so, for example, if we look at the mass of nutrient assimilated by the crop over the mass that's applied, the maximum amounts that are currently you can find in the literature are 50% and 25%. So if we could increase that, for example, by say 20%, and maybe that's a lot, right? But at 20%, we could save 3.5 billion kilograms of nitrogen per year, and there's a huge economic savings for that. So even if we got to 10%, half of that is, again, a huge economic and also resource savings. And so there's a huge potential here to have a positive impact. And so if we focus our attention on farm and trying to improve nutrient use efficiency, there's a number of opportunities and different technologies that are being developed in our lab, in others' lab. And so the question, I'm not going to talk about those technologies today, given the topic of my talk, but what I will talk about is evaluating the technologies that are being developed, specifically the use of nanomaterials to enable environmental and economic savings, and trying to do this without compromising performance. And there are two review articles, this one in Nature Food and this one in Nature Nanotechnology, that talk about a lot of the opportunities. And so I will direct you to those two articles in order to see the plethora of nanotechnologies being talked about and developed. But what I'm going to demonstrate is sort of how we use the systems perspective to evaluate nanomaterials early and identify and direct the design so that we can avoid that regrettable substitution, right? So thinking about how back at the beginning, I was talking about the corn-based ethanol and the chlorine and the compact fluorescent light bulbs, right? So there are these unexpected implications once we get the technology is mature and adopted, and we want to try to avoid as many of those and all of them if possible. And so when we are developing nanomaterials for use on farm and for crop production, we want to consider which nanomaterial, so we have a wide range of compositions that we can think about, and then also how we design them and make them. So that's synthesis, which synthesis we use, which shape, which size we want to adopt. And then for the example that I'm going to show, I'm using nitrogen offset, so offsetting the nitrogen input, but you could easily run these same analysis for phosphorus offset. And the three examples in this particular study are soil amendments, foliar sprays, and seed coatings. And I'm only going to show the seed coating example just to demonstrate what we did. And so with seed coatings, what you see here on the vertical axis is an increase in germination. So that's the performance metric with seed coatings that we're concerned with. And this is a percentage. This is an increase in germination. So 0% up here to 16%. Along the horizontal axis, the environmental impact that we were concerned with was embodied energy. And so cumulative energy demand, or embodied energy, is oftentimes used as a surrogate for environmental implications. It's a fairly straightforward one to calculate, and we have a lot of information on it. And so what we looked at first was, we want to benchmark our nanomaterials to conventional chemicals. And so what you see here are 4 different conventional chemicals that are used to help germinate seeds already. And then what we did is we took information from all of the studies that currently are in the literature on nanomaterials that were being applied to seeds, and looked at the performance that they reported. The performance increased, right, in germination compared to the control, and then calculated the embodied energy of the nanomaterial. That includes the synthesis, so which synthesis was used, how much of the nanomaterial was actually used on the seed to realize the benefit. And what we did is there's a number of different syntheses for each nanomaterial type. And so we included all of those. And so that's what the range here on the horizontal axis is from. And so what this particular analysis of seed coating tells us is that nearly all of the nanomaterials have a performance enhancement in the germination of seeds. And then in particular, the nanocopper and zinc oxide, these 2 particular designs of those compositions are really have a high potential to have a positive impact, because they not only significantly increase the germination rate, they also have a lower embodied energy compared to the 4 conventional alternatives here. So not only are we increasing the performance, we're also reducing the environmental impact from the upstream production of these nanomaterials. And so what did we learn from this systems analysis is, the type of nanomaterial matters and how it's synthesized, right? So the type, right? So here, copper and zinc being the ones that are most promising. But then the range here in all of these is indicative of how they're being synthesized. And so we ideally want to synthesize using the lowest embodied energy. The crop type and the location of the application should be considered. So that's not obvious in this particular plot, but the each individual study for each individual data point has used a different crop, and those crops are going to be applied, grown in a variety of different locations. And so we need to think about that. We can quantify other environmental metrics. So like I mentioned, cumulative energy demand is often used. There are a lot of other environmental metrics that are available to us that we can take the same approach, and just essentially what we would be changing here is the horizontal axis. So we could look at things like eutrophication potential and a human health endpoint, global warming potential, things like that. So as I mentioned in this study that I talked about, we also looked at foliar application, we looked at soil applications, and we looked at nutrient recovery. Separately, we've looked at nutrient recovery, and we've also looked at sensing for chemical and biological analytes. So there's a variety of different applications that you could apply this approach to. And some applications are going to have greater potential for net benefit than others. So for example, I showed you one that was really promising here. In this same study, the soil applications did not have support a beneficial use of nanomaterials compared to the conventional alternative. There's a higher cumulative energy demand to their use. And so thinking about which applications we use nanotechnology for is something that we should be cognizant of moving forward. It's not going to be the silver bullet for every single application. And so with that, I welcome your questions. I appreciate your attention, and have my contact information here in case we don't have time to answer all your questions. I'm happy to engage in conversation with anybody out there who's listening. So thank you very much.
Thank you very much, Professor Gilbertson, for the nice presentation. Mm-hmm. session is open for questions. And let's see if, okay, one from Abdullah Karoum. Thank you, Professor Gilbertson, for this interesting presentation. What about the interaction between the phosphorus and nitrogen with other macro and micro elements in the nutrient efficiency point of view? Let's say I was seeing.
So I guess I'm going to interpret this question as under the current status quo of delivering nitrogen and phosphorus to the environment, and perhaps related to the efficiency of the system calculation. It's uncertain, but we did not take into consideration. I don't know. Can you help me with understanding? I'm not exactly sure what we're asking here.
Well, I think
is this related to the nanotechnology, or
I don't know, maybe it's related to the when we are combining both. Is that has an effect on the let's say the efficiency
calculation?
Yes.
Yeah, so I think that this is inherent. So for the nutrient use efficiency, I'm not sure that you can still see my slides, probably not. These values right here in the on-farm, the 50% and 25%, as far as I know, are already considering, for example, that phosphorus gets bound up into soil and is not taken up by the crop, and that the nitrogen will undergo nitrification and denitrification processes, and for example, to get it into the form of nitrate for taking up, for the crop roots to take up the nutrients. And so the 25% and 50% that we used are the maximum of that range, and that those interactions are already incorporated into that value.
Okay, I think we can take another question. Well, thank you, Professor Gilbertson. Do you think that different technique of application of the nanomaterial would have the same impact on nutrient uptake?
Yeah, that's a great question. I'm interpreting this as, should we shift from, I guess for fertilizers, broadcasting sort of indiscriminately over soils versus more of a targeted delivery, for example, through fertigation processes, perhaps. Same with pesticide delivery. If we spray the pesticides indiscriminately versus trying to apply them specifically to the leaf to reduce the waste. So most certainly, when you change from an indiscriminate to a more directed delivery, you're going to reduce the you're going to increase the efficiency, reduce the loss, and so that's something that we can do. But that's something that's available right now to farmers, and they're not adopting these approaches for large-scale production, for example, of corn, which is a common model crop that we use in our thinking, because so much of it is grown and so much of the inputs are applied. And my sense is that it's because it takes a lot more time and therefore costs a lot more. They might need different tools and equipment in order to apply it more directly. And so if nanotechnology were able to leverage the existing application approaches without changing the farmer's behavior, but direct the actual nutrients and the pesticides to the target, like Francois was talking about, then I think that that's a real opportunity. And I think that we can, based on some of the literature that's out there that Francois highlighted.
Okay. Thank you. I would think this is the last question to move on from Hisham Fanniri. Great presentation. Thank you. How can we assess potential downstream toxicity to animals and humans, if any?
Yeah, so the if any, there most certainly is a concern over this within the field. Absolutely. And there's a whole body of literature on nanotoxicology that we need to be aware of and incorporate this into our thinking. And it's also very astute of you to notice that everything that I've done so far is from cradle to gate, so how to manufacturing of the nanomaterials. We don't have a lot of information right now to know at the field scale, even at the lab scale, how much of the nanomaterial is taken up by the plant, where is it going? If we're using a nanomaterial that doesn't degrade, where does it accumulate in the plant? Is it accumulating in somewhere that humans or animals will consume? That's of concern. If it's not being taken up by the plant and it's just a carrier, is it does it remain in the environment? Does it leach to the waterways? And what does that mean for things, for example, like copper, which is an antimicrobial, and what are the ecological effects of that if we were to have a huge concentration build up? The tool that I use, life cycle assessment, cannot right now directly, I'm sorry, I'm getting feedback, so it's hard to say. The life cycle assessment cannot directly quantify those impacts that you're talking about, but it's something that the community, the life cycle assessment community, is currently working on updating the models to be able to account for these things. So it's a great question. Thank you.
Thank you very much again for the nice presentation. And we can move now on to our third speaker of the session, who is Dr. Juan Pablo Geraldo. I will just give a small introduction. After receiving his PhD in plant biology from Harvard University in 2011, Dr. Juan Pablo Geraldo worked as an NSF postdoctoral fellow on plant nanobiotechnology at MIT. Since 2015, Dr. Geraldo lab at the University of California, Riverside works at the interface between plant biology and nanotechnology. His lab aims to develop nanoparticles-based research tools to study and engineer plant function at levels of organization ranging from organelles to tissues and whole plants. Dr. Geraldo, the stage is yours.
Thank you for the nice introduction. Can you guys hear me well?
Yes.
Okay. And thank you, Francois, and the organizers for inviting me to speak at this conference. My lab at the University of California, Riverside, as you mentioned, has been focusing in the past 5 years to focus on using nanotechnology-based tools for both for fundamental understanding of plant biology, but also for applications, such as the one that we are talking about here, improving agrochemical delivery, and also for monitoring plant health using nanosensors. And many of you already have touched upon these topics. I've seen some slides that highlighted this recent review in Nature Food, in which we highlight opportunities, challenges, barriers for applying nanotechnology to agriculture, and including, for example, topics that I'll talk today about how we can use nanosensors for monitoring plant health, whether a plant is stressed or is suffering from a nutrient deficiency, such as phosphorus. How nanofertilizers can be help us to more efficiently deliver those nutrients to the specific sites in plants where they are needed to be assimilated into the plants without being lost into the environment. Or other tools that I won't be talking about, like genetic engineering, like delivery of DNA, genetic elements, and RNA for improving crop improvement and developing a new and more tolerant or efficient varieties of plants, and nanopesticides, and among others, and many more that I will not have a chance to talk about today. So I look into what can be done today to image the status of phosphorus in a plant. And I came across this very interesting article in Plant Physiology published just this year. They use a technique based on chlorophyll fluorescence, in which you can use chlorophyll fluorescence imaging camera called an imaging PAM, and you can detect levels of phosphorus, in which like red that you can see here means like higher phosphorus, and green means deficiency of phosphorus. And it turns out like that correlates, that measurement, which they call MPQ, which is a technical term called non-photochemical quenching of chlorophyll fluorescence, is correlated, for example, with the phosphorus in the plant. When these values are around 0.5 or low, then you get, this means that the plants have a good level of phosphorus. And when this MPQ value is high, that means that you have a lower level of phosphorus. So this is an interesting technique that shows that like we could potentially start imaging the status of a nutrient in plants, in this case in barley. However, it still doesn't measure yet the key molecules that are interesting here, nutrients directly. It's an indirect measurement based on chlorophyll fluorescence. So nanotechnology offers the opportunity to directly sense chemical signals or molecules in plants that are associated with plant health. For example, we have demonstrated in our previous work that by embedding nanosensors in spinach plants, we can translate the levels of a chemical analyte present in groundwater that is transpired from the roots up into the leaves, modulating the fluorescence of these sensors, as you can see here, from like high intensity signal to a much lower signal that is communicated to a near-infrared camera that is could be available. That's part of available equipment for precision agriculture or a type of another agricultural phenotyping facilities, or even as we demonstrated, it could be a camera from a smartphone that you can use to connect to the web, and this plant can communicate via an email or a text signal, like the status of the level of these chemicals in the environment or plant health. And this is the vision that we have for using nanotechnology in plants as sensors. Can we translate the internal chemical signals of plants into signals that we can measure like optically? For example, this is another application from my lab in which we monitor glucose using some particles called quantum dots. Glucose is the direct product of photosynthesis in the chloroplast. And so we directly measuring photosynthetic output using this tool in which the quantum dots interact, that we made by coating them with phenolboronic acids, interact with glucose, and when they change the aggregation state, they can modulate their fluorescence inside leaves and report the status of glucose in the plant. So you can see it again as a direct way to measure photosynthesis, as opposed to the metrics that we have now today based on carbon dioxide assimilation or chlorophyll fluorescence. And I summarize the opportunities, challenges, and future potential work done in this field in a review that was published in an issue about nano-enabled agriculture last year in June 2019 in Nature Nanotechnology. In that review, we showed how we could potentially use nanosensors such as like nanotube-based sensors, quantum dot-based sensors, wearable sensors, or even nanotechnology-mediated genetically encoded sensors and others that are currently in development to translate plant chemical signals that are associated with plant health, including stresses like drought, temperature, and in this case, for like purpose of this meeting, nutrients like nitrogen and phosphorus, and or phosphate. So like that is something that we are interested in pursuing in my lab, and I will be discussing how we can use these nanosensors to translate chemical signals related to these nutrient deficiencies into digital information, particularly into optical signals that can be recorded by electronic devices like hyperspectral imaging cameras that are used in phenotyping facilities, or plant screening systems, or autonomous manned terrestrial and aerial vehicles, and why not maybe someday using our own portable devices like smartphones for different range of applications, which include like laboratory research, or like from laboratory research to urban farming and precision agriculture. And so how do these sensors work? So this is an example of one of the like first sensors that we have been working on, mainly for purpose of research, so that we using carbon nanotubes that are coated in a polymer that impart them selectivity to molecules that are very difficult to detect. For example, hydrogen peroxide and nitric oxide. This is a very small molecule with short lifetimes, and these nanotubes are able to, nanosensors are able to modulate the emission of their fluorescence intensity and report it to a near-infrared camera and the form of these NIR images. And with that optical information, we can detect with high spatial and also temporal resolution changes in the status of these particular chemicals. And in my lab recently, we use a hydrogen peroxide-based sensor to monitor plant health in this way. So hydrogen peroxide is a molecule that tends to accumulate in plants that are not healthy, that are suffering from stresses or potentially nutrient deficiencies. And so when we insert these nanosensors into plant leaves, we could monitor with high sensitivity within physiological range of between like 10 micromolar to even 1 millimolar changes in hydrogen peroxide as we are adding and removing it to the leaf surface. And we can monitor with a near-infrared camera like these changes in the status of hydrogen peroxide in the plants with good temporal resolution, in which the sensor shows that like the fluorescence intensity quenches, is reduced by about 15%, and then it increases and recovers after we remove hydrogen peroxide. So we asked the question, can we use hydrogen peroxide as a proxy to monitor plant health and test and basically identify if the plant is stressed or healthy by using this imaging technique of nanosensors? And we test the different stresses, UVB light, highlight, pathogen stress, and wounding, and found that like for the UVB light and highlight and pathogen stress, as you can see, the sensors report decrease in fluorescence associated to the beginning of the stress, UVB light, highlight, or pathogen stress, in a way that in several cases, particularly in the pathogen stress, is reversible. So that means that we can continue using this sensor for monitor plant health over multiple periods of time. Interestingly, like wounding, like when we cut the leaves or excise the leaves, didn't cause any changes in the sensor fluorescence. And so that means like other stresses, like for example, a herbivore breaking leaves or eating leaves will not be able to be detected by this technique. So, we're moving forward now to with a collaboration, Sebastian Cruz Lab in Germany, to also make these types of nanosensors with materials that could be more easily biodegradable and sustainable in the environment. Like carbon nanotubes are like not easy to degrade, and so that's something that we are mindful of, like translating these technologies into more sustainable materials made of silica, for example, nanosheets that we have discovered like there can be 10 times brighter than the single wall carbon nanotubes. As you can see in this image, these are the carbon nanotubes, and this is the silica nanosheets. In this near infrared fluorescence image, you can see the differences in brightness with the same excitation. So this could allow for like potentially simpler sensing with higher signal to noise ratio imaging with an LED, simply with using a system having an LED excitation and camera, perhaps like not even a near infrared camera. It could be just a normal silicon dioxide-based sensor camera. And so we demonstrated that in Arabidopsis plants, like we can image these silica nanosheets, and we're currently working on applications of developing these silica nanosheets into sensors. Now, moving forward, talking about like how to improve the agrochemical delivery using nanotechnologies, the second part of my talk, I saw Francois did a great introduction to this area, and actually even I remember seeing this slide here, so I won't talk much about it, but my lab has been working on the area of a more efficient and targeted delivery of the nanomaterials with agrochemical cargoes into plants. And the goal is like we can, as Leanne discussed, like improve the efficiency of the application and reduce the losses in the environment. And there are all these questions around like, can this be scalable? Can we reduce the environmental footprint? Is it going to be socially acceptable, and is economically viable? And I'm currently working with an industry partner on these questions, and I kind of talk more about that project. It's something that is still in the works, but the answer to that is like we think so. Like there are ways that we can make these technologies to work, but we need to be mindful, as Leanne pointed from the beginning, in designing materials that not only can be targeted nutrients, but also comply with all these other requests that in order to have an impact in the field and of agriculture. So with that in mind, my lab has been looking into ways to use more benign, sustainable nanoparticles like carbon dots that are made of organic chemicals found in plants. They're made from basically urea and citric acid. They have a carbon oxide nitrogen core that can be biodegradable. And these particles are fluorescent, so they allows us to track it inside of the plants. And as you can show you, like we can start coating them with modifying their surface. We can control better where do they go, and the properties like size and charge. We can modify where they go exactly into the plants. And we are looking into applications that are non-dependent on the type of soil, but instead only on the type of the plant, because we think that we can control better, like design a better nanotechnology-based tool if we know that what exactly will be the biological matrix that these particles will be interacting with. In this case, we've been working on cotton leaves and also corn leaves. As Leanne mentioned, corn is one of the major crops here in the United States and worldwide. And so we've been discovering nanoparticle formulations that allows us to deliver these carbon dots through different pathways, through the cuticle and through the stomata, into a specific subcellular compartments like chloroplasts. And so chloroplasts are actually one of the places where you want to target phosphates. These are the sites where phosphates are assimilated in the plant and converted in ATP. So it is mitochondria too. So these are major sites where you want actually that phosphates to be delivered. And I'll show you what properties of the particles allow you to do that. And so these are some models that we created based on several years of work with multiple particles, not just carbon dots, but also cerium oxide and silicon dioxide nanoparticles, in which we identified that the charge, setup potential here of the particles, and size is crucial to determine the efficiency of delivery to the chloroplast that you can see here in these confocal images of leaves shown in magenta. These are chloroplasts, and green are the particles that overlay with the chloroplast. So we discovered that in this region where high setup potentials, positive setup potentials, between 10, 15 to 40 millivolts, and below this size cutoff limit about 15 nanometers is where we have the highest efficiency for delivery to particles to the size of assimilation of phosphates in the chloroplast. And for maize, it was a little more restrictive, but it was something that we found that the positive particles that are more highly charged could be delivered to the chloroplast if they have this setup potential and this small, relatively small size. And another way that we can do targeted delivery, not only by controlling size and charge, is by delivering, by coating the nanoparticles with targeting peptides and molecular recognition motifs. And this is something I'm very excited working in my lab, where we want the plant to guide the particles to specific sites, to where the nutrients or chemicals are needed. And so the plants already know how to do that. They use, for example, these in chloroplasts, these sites of assimilation of phosphates. They use translocon channels to deliver proteins to make the chloroplast. So we are kind of hacking into that molecular biorecognition machinery and coating the nanoparticles with peptides, these chloroplast peptides, for guiding them more specifically to where we want them to be. And with that, also, we can coat the nanoparticles with molecular baskets like these beta-cyclodextrins and load cyclodextrins and load chemicals. Like this could be herbicide or pesticides, but we can envision loading other types of chemicals potentially for agrochemical delivery and we deliver with high specificity these cargoes into the sites to where I need it. And I'm not going to have time to talk about this, but this type of targeted delivery by peptides could also be applied for other types of nanoparticles. Perhaps you have heard carbon nanotubes have been used now for delivering DNA, and also I just discussed as nanosensors. So this technology could apply to many types of particles. And so how does that work? So we have used fundamental research, we have focused on Arabidopsis plants because we understand very well their molecular biology. And we have also used quantum dots that are made with cadmium and telluride because they allows us to both track the fluorescence of the particles in the plants and also detect them by other advanced analytical tools, as I show you, like elemental analysis and others. And with that, you can see here that the quantum dots coated with this chloroplast targeting peptide that are shown here in green co-localize very well with the chloroplast and shown here in magenta. This is like a high level of co-localization that we have not seen before. And when we randomize the peptide, we do not see the same level of co-localization at all, indicating that the sequence of this peptide is important. And when we don't coat the particles with the peptide, we don't see that same level of co-localization. In fact, like without the peptide, you have about 40% co-localization, 40% of chloroplasts contain nanoparticles that we want to target, whereas like with the targeting peptide, about 80% of the chloroplasts contain those particles. And so, as I mentioned, like the quantum dots for fundamental research allows us to also track, prove that we are actually delivering these particles into chloroplasts by other advanced analytical tools. So we extract the chloroplasts from the plants, isolate them, we look at their fluorescence here again, and we find that they are co-localizing, again, the particles with the chloroplasts. But then we also measure the cadmium and tellurium content and find that those chloroplasts that were targeted with the quantum dots in the targeting peptide have much higher levels of cadmium and tellurium, indicating that the particles are indeed inside the chloroplast. And this targeting peptide, now moving more towards like applications, can be found, it's conserved across many dicotyledonous plants. Several of them are like crops. So I just talked to you about a demonstration that we published in Nature Communications using Arabidopsis thaniana plant model system. But as you can see here, like this same sequence of targeting peptide that we took from a Rubisco, basically a small subunit that plants use to deliver those peptides and precursors of proteins into the chloroplast, is also found with a very similar sequence in crops like soybeans, for example, or tomatoes, or grapevines. And we have identified another sequence that we need to work on more on that applies to monocots, like cereal grains and large like crops, like the crops that have a much wider global impact, like corn and wheat and so on. And so I mentioned earlier that not only by coating the particles with a targeting peptide, but also by functionalizing, adding a molecular basket, we can load agrochemicals and more specifically target to the sites of interest. So we, for example, in this paper, loaded the molecular baskets with methylvilagen, which is also called paraquat, a herbicide, and shown that we can more specifically deliver this cargo into the chloroplast. Once this chemical MV is delivered into the chloroplast, it produces reactive oxygen species like superoxide that we can measure with fluorescence dyes. So this is an ROS dye here that measures like the intensity of the ROS levels as response of adding the targeted delivery of MV versus adding the MV alone. And as you can tell from the images here, the overlay, there's a high level of specificity of generation of ROS using the targeted delivery approach versus just delivering the agrochemicals alone. In fact, like we find based on this assay of fluorescence intensity of ROS, that we can improve, almost like double or triple the efficiency of the delivery of this cargo using nanomaterials compared to chemicals alone. So this to wrap up, I hope like I've given you some evidence of the different types of applications that we can use for monitoring plant health and also delivery of agrochemicals in plants with more precise and more in a way that is guided more by the plant itself, not by like us as humans. We determine when the plants are healthy, when the plants are sick. We determine when we apply agrochemicals in the field. But in this case, what I think see the potential of nanotechnology is that the plants themselves can tell us when they are when they need to have agrochemicals like nutrients and phosphates delivered into the plants, and also using their own molecular recognition machinery, we can target the delivery of nanoparticles by tuning their size and charge, and also adding these guiding peptides that hopefully improve the delivery efficiency from what we have right now. Phosphates, I heard earlier, something like about 25% use efficiency, maybe potentially double that. And so something that looking forward to discuss with this session and others after this meeting. And with that, I just want to thank my research group. We are a team made of like plant biologists, chemical engineers, material science, and chemists working at the interaction of nanotechnology and plant biology. And we're supported by several funding sources, including NSF and USDA. And so with that, I just want to thank you again for the opportunity to discuss my research in this session, and I welcome all your questions.
Thank you very much for this really inspiring presentation. It was very, very exciting, let's say, talk. I enjoyed very much. Now we can start, I think, with some questions. I think there will be some. Okay, from Abdurrahim Ben Zawiya. A recent study confirmed that bees detect a magnetic field emitted by plants. Can it be a perspective application for nanotechnology?
Well, it's interesting you mentioned that, because with the Center of Sustainable Nanotechnology that I work with, we are working on some nanoparticles that are able to detect very small changes in magnetic fields. And that is something that I find fascinating. It's a topic that I don't tend to think as much, but I think nanotechnology can be used to sense small changes in magnetic fields in plants. And in science, we have seen that when we discover new tools, often that leads to understanding new discoveries in understanding of the biology of organisms. So yes, there is an opportunity to sense magnetic fields with nanosensors, but that's not out there yet, but stay tuned.
Okay, I don't know if there is any other questions from Abdullah Karoum. Thank you, Professor Geraldo, for this nice presentation. I'm wondering how plants react with the nanosensors inside their cells. How do cells recognize these nanosensors?
This is an excellent question. I think you guys are already knowing what I'm working on, because you're asking me all the questions that we're currently addressing. So my group is actually working right now and is very interested in looking at how nanoparticles, not just nanosensors, are coated with biomolecules when they enter plant cells. So these coatings of biomolecules are called coronas, and it's an important field in the area of chemistry of nanomaterials. And by changing the corona or the coating of a nanoparticle, you change their identity and potentially where they can go into the plant. They also can change their function. So what we're aiming now to do is we're delivering particles into, again, Arabiopsis plants that allows us to, because it has a lot of databases for proteomics, lipidomics, and genomics analysis to identify which are some of those key biomolecules, and then determine how they interact or react with plant cells. There are, on the more application side, what I have seen is and many of my colleagues have seen is concentration is important, and also transformation, where the particle dissolves and change into other elements that can cause potential side effects in the plants. And so that is something that we always keep in mind when we're doing studies for agriculture. And that's why I think we need to choose carefully the materials that we're working on. So that's why we're moving into working more with carbon and based silicon nanoparticles made of elements that are more environmentally friendly and sustainable.
Thank you very much. I think we take this one as the last question because we are ahead a bit of time. Good presentation. Thank you. How can nanotechnology help to improve the sensitivity and selectivity of smart plant nanosensors?
I think, look, what I found fascinating about this area of nanosensors is that the plant itself can communicate to us whether it needs the nutrients or not. If you think about the way we address agriculture now, is like farmers or drones or some other machines like are assessing the status of the plant. I would like the plants to tell us like whether or not it needs the nutrients or whether or not it needs to be treated for promoting its health. And so hopefully someday we will accomplish that. And that's how nanosensors will be able to help by translating the language of plants is that chemical signals that we don't understand. But if we can translate those chemical signals into digital information, computers can decode that, and we can hopefully someday communicate with plants in that way.
Okay, thank you very much again, Dr. Geraldo, for this inspiring presentation and for your presence. And then I will move now to the next or the last speaker of the session, who is Professor Mohammed Al-Hsini. He received both his master and PhD from the University of Bordeaux in France. After postdoctoral research with Professor Bernard Jussom, he started his academic career at Qadi Ayyad University in 1995 as an assistant professor, and then in 2002 he was appointed full professor at the same university. He was during several years a visiting scientist in different institutions around the world. In 2013, he was the recipient of the Alexander von Humboldt Research Award for Experienced Researchers in Germany, very high distinction. His research covers many aspects: organometallics, macromolecular chemistry, heterogeneous catalysis for fine chemical synthesis, and homogeneous catalysis in biopolymers and nanocomposite materials. Green chemistry, including the chemistry of biosourced materials. Professor Al-Hsini, the stage is yours.
Thank you very much for this introduction. First of all, I would like to give my warm thanks to our organizing committee, and my special thank go to Abdullah Karoum to invite me and to allow me the opportunity to present a part of my work during this workshop, in which I will talk about biodegradable materials based on polycaprolactone from synthesis to coating fertilizers for slow release. Why we are interested to these kind of materials for slow release and even for coating packaging? As you know, the most useful materials for coating fertilizer is based on conventional plastics, and synthetic plastics. Its conception and production, it's growing year after year. And for example, the annual production of plastics in 2018, it was around 359 million tons a year, instead of only 2 million tons in 1950. This is because of low cost, of course, and also because of excellent properties of plastic, like barrier properties, mechanical properties, and also low density. All these properties leads to application of plastics in agriculture, in packaging, biomedical, automotive, and construction. But however, the main source of a plastic is performed with non-renewable resources, oil. And most of these polymers are not biodegradable. Their accumulation in nature causes a serious problem for the environment for every country in the world.
Sorry, Professor.
Sorry?
Sorry, Professor, just if you can put your slides on full.
Yeah, I'm sorry. That's okay?
Yes, perfect, yes. Thank you.
The main source of a plastic is petroleum, which is non-renewable, and most of these polymers are not biodegradable. Their accumulation in nature causes a serious problem for environmental for every country in the world, and this material waste now occupy a large volume in landfills and dams, and now we start to talk about plastic continents. And to overcome to this problem, attention has focused to biodegradable polymer. And when we talk about biodegradable polymer, there is two kinds of bio-based polymer. It's like polysaccharide, cellulose, chitosan, and others. And we can also talk about synthetic biodegradable polymer. The synthetic biodegradable polymer can have the same properties like polypropylene or polyethylene or polyacrylate, because they are thermoplastic polymers. And among these polymer, we have aliphatic polyesters, mainly polylactide, polycaprolactone, polyhydroxybutyrate, and their copolymers have attracted considerable attention because they have a potential application in agriculture, transportation, packaging, bioindustry, and also in electronics. And to prepare this kind of polymer, there is two kinds of methods. There is polycondensation reaction, which is less expensive, or Reynolds polymerization, which is much more expensive. But the first, the polycondensation reaction, leads to only broad molecular weight and low molecular weight with broad dispersity. This is not possible. This is allowed to this polymer to not have any application for industry because low molecular weight. But Reynolds polymerization, we can prepare a polymer with high molecular weight. We can control the molecular weight by controlling the ratio between monomer to catalyst, and also we can achieve specific end group in the end of the polymer, and we can prepare block and multi-block polymer. The main method to prepare this polymer, or this is the Reynolds polymerization, but the catalyst that can be used, this is metal alkoxide based on aluminum, titanium, tin, zinc, iron, and bismuth. But the most useful catalyst, even for academic research or industrial production, is based on tin metals. For academic research, tin alkoxide is the most useful catalyst because butylated tin 4 alkoxide are cheaper, commercially available, thermally stable, and they lead to the really good control of the polymerization. We can prepare block, multi-block polymer, high molecular weight. We can achieve specific end group. But all these complexes, even if they are excellent catalysts, they cannot be used in industry because this tin carbon bond, which is really toxic. But the standard catalyst for Reynolds polymerization and other technical production, for example, of polyurethane, is based on tin octanoate because this compound is admitted by American Food and Drug Administration as food stabilizer. But for some reaction or to increase its activity, we have to use the alcohol as co-initiator to increase the activity of the catalyst toward the Reynolds polymerization and the alcohol can act as transfer agent during the polymerization, and consequently we lost the control of the polymerization, which is very required to have really a good polymer with specific properties. In our group, a few years ago, to benefit from the excellent activity of butylated tin 4 and low toxicity of tin octanoate, we developed tin 4 alkoxide. Why? Like butylated tin 4 alkoxide, we don't need to use any alcohol as co-initiator. And like tin octanoate, with water dilute to tin oxide, which is admitted by American Food and Drug Administration, and we test this catalyst with, of course, we compare their activity with standard catalyst tin octanoate, and we can observe here that we have really high activity compared to the standard one. And we use it also to prepare block polymer, polylactide, block polyethylene caprolactone. We start the polymerization first with polylactide at the end of the conversion, and we add ethylene caprolactone. And as you can see, we have two lines. The continuous line is the first polymer, and the discontinuous line, this is a block polymer. As we have monomodal curves, this indicating that we have polylactide linked to polyethylene caprolactone, we have graphs. But this complex, even if instead of high activity of this complex, it cannot have any application in industry because it's really sensitive to the moisture, and to use it as catalyst, we should work in glove box or in schlank line, which is not easy in technical production. For the same way, in the same aim to develop really simple catalyst to prepare high molecular weight polymer, we investigate this tin 4 compound. As previously we showed that this compound leads to tin oxide by reaction with water, and this compound leads to tin alkoxide by reaction of alcohol. We test this complex with alcohol to polymerize the lactide in the same condition than before, and we observe a really good control of the polymerization. But what I would like to show you in this table, we use this catalyst without using any solvent in bulk condition at high temperature, at the same temperature than in industrial preparation of polylactide, and in bulk without using any solvent, but with high ratio, really high ratio, 10,000 to 1, 9,000 to 1, and 5,000 to 1. And we observe really high molecular weight, and the total conversion were achieved after 15 minutes. It was the first example in literature in which we can prepare polylactide with catalyst in open air, because all other catalysts we should to work under argon or nitrogen or vacuum. So we have now a good catalyst. We prepare high molecular weight of polylactide or polyethylene caprolactone. And the aim of the application, it was the packaging or coating fertilizers. But this aliphatic polyester presents some limitation. They present low thermal stability, low barrier properties, and low mechanical properties. And also they are highly hydrophobic for coating material, for coating fertilizers. The hydrophobic character sometimes presents some drawback. Then we should to improve these properties to use it for packaging and also for coating fertilizers. And for that, it was reported that incorporation of fillers like polysaccharide or clay with the polymer increase their thermal properties, their mechanical properties, and also these barrier properties. But to improve these properties, we should have really homogeneous dispersion of fillers inside of the polymer. And there is two main methods to incorporate the nanofillers with the polymer. Melt blending of polymer with fillers. This is really simple method. We melt the polymer, we added the fillers, we mix, and that's on. We have our nanocomposites. Or we mix the monomer, catalyst, and monomer, catalyst, and fillers, and we grow the polymer inside the filler with the fillers. The first method, it's really simple, but it's convenient when the fillers and the polymer are the same character, both hydrophobic or both hydrophilic. When one is hydrophilic and the other one is hydrophobic, we will have really difficult to have a good dispersion of the fillers in the mono. It's like if you would like to mix the oil with water. But the in situ polymerization of monomer with fillers, it's really good method. When, for example, we would like to polymerize the monomer with radical polymerization, because for example, filler with OH group, it doesn't affect the radical polymerization. But in our case, we have a polymer. Our polymer is aliphatic polyesters. Our fillers is both polysaccharide or hyalosite nanotubes or clay. Both are hydrophilic. Normally, we melt, mixed, and we should have a composite with the good properties. But in our case, it was really difficult to get this, as our polymer is hydrophobic and the fillers are hydrophilic. It was really difficult to have a good dispersion, a good addition between both. Then we opt for in situ rain-opening polymerization for epsilon caprolactone or lactide with filler. But when we checked the literature, it was really hard for us with standard catalyst to get to high molecular weight grafted on the surface of polysaccharide or clay, because the OH group, more we have OH group on the surface, more we have some transacidification, and it was reported that it's really difficult to get a high molecular weight with standard catalyst. For that reason, what we thought, why not to use our catalyst as we success to prepare polylactide in open air without purifying the monomer. For that, we used our catalyst with polysaccharide like GoArgom and mineral clay hyalosite nanotubes. And as both present some OH group in the surface, The OH group can play the role of initiator, and first we grab the catalyst on the surface, and that we start the polymerization. The reaction, it's easy. We mix the filler in open air with the catalyst, and we used bulk polymerization. We use the monomer as solvent too. At room temperature, we have a reaction between the fillers and the catalyst. We graph the catalyst by formation of tin alkoxide group, and at 120, we start the polymerization, and really we graph the polymer on the surface of the filler. Of course, we have some free polymer because we have some water with fillers. As we can see, we have, this is a net polymer. We prepare it without using any fillers. We have around 98,000 Dalton, and with 1% we have almost the same, even with guar-gum or with hyalosite nanotubes. But the molar mass decrease by increasing the contents of the filler, even with guar-gum of hyalosite nanotubes. This is normal because we use the same quantity of monomer. And when we have 1%, like if we have 2 OH groups, so we divide, we have only 2 chain growing on the surface. When we have 3%, we have more chain growing, and when we have 5%, that's the reason why the molar mass decrease. But what we observe is that when we increase the contents of fillers, the reaction is really fast. For example, with 5%, we need only 10 minutes, but with 1%, we need between 15 to 20 minutes. And to prove that we really will graft the polymer on the surface, we analyze first the nanocomposite by IR. As you can see here, we have polycaprolactone, IR spectrum polycaprolactone, hyalosite nanotubes. That one, this is hyalosite nanotubes, CRAD hyalosite nano before purification, and here after purification. And we can see even with hyalosite nanotubes or guar-gum, we have the same bond we have carboxyl group between 2000 to 2050, and we have OH group on the surface. That's indicating that we really graft the polymer on the surface of fillers. And to confirm that really we graft our polymer on the fillers, we studied the suspension stability of our nanocomposite in dichloromethane and the mixture between free polymer and fillers in dichloromethane, and also fillers in dichloromethane. And as you can see here, after a few hours, we observe the precipitation of fillers. And with our nanocomposite, we still have stable suspension even after 24 hours. This indicating that really we graft the polymer on the surface. The aim of this study is to improve the mechanical properties, and we can see the micro hardness that we improve the mechanical properties even with guar-gum or hyalosite nanotubes, and also as the balance between hydrophobic character and hydrophilic character. It's crucial parameter for coating fertilizer and also for food packaging. So we studied the contact angle of our nanocomposite, and we observed that the contact angle decreased by increasing the contents of the polymer. This is indicating that we can organize our materials if we need more hydrophobic or more hydrophilic just by varying the ratio of the fillers. The same image show us that we have really good dispersion of the fillers in the matrix. This is with hyalosite nanotubes and even for guar-gum. And the incorporation of the fillers with the polymer increase the thermal stability of nanocomposite. As you can see, even with guar-gum or with hyalosite nanotubes, really we improve the thermal stability of the materials. But for biodegradation of materials in soil, we observe that more we have fillers, fast the degradation of material. This is because the fillers, it's hydrophilic, so it absorbs water. And as we have aliphatic polyester, water reacted with ester, so we hydrolyze, and facilitate its decomposition or biodegradation. So we have a good materials. Normally, it's intended for packaging and also for coating of fertilizers. As the workshop is deal with agriculture, I will talk about coating fertilizers with our material. But first, I would like just to tell you why we interested this, because as you know, the population is growing year after year. We have to satisfy the need of food for the world population. We use enormous fertilizers, and the most used fertilizer is water-soluble fertilizers. And these water-soluble fertilizers, unfortunately, it releases fastly all nutrients that it's not possible for crops to uptake all these nutrients. So we lost a lot, and we pollute water, we contaminate water, and we have some evaporation, and consequently we have to use frequently, and it costs more expensive. And one solution for that is, why not to coating fertilizer to control and the slow release of the fertilizer in the soil. In the literature, the first material which has been used for coating fertilizer was based in sulfur. These materials show really good slowing of the fertilizer, but it presents some poor mechanical properties, and sometimes because not irregular slow release of fertilizers. But the most efficient polymer or coating materials for fertilizer are based on synthetic polymer, like polyacrylate, for example. They lead to the good control release. They read and slow release because of high barrier properties, high mechanical properties, and also for hydrophobic character. But unfortunately, this material is not biodegradable, and after fertilizing, its accumulation in soil degrade the quality of soil. And it was reported that we did about 50 kilograms per hectare is accumulated in soil every year when we use this kind of type of fertilizer. How to overcome this problem? One solution is to use bio-based polymer or biodegradable polymer. And the most or the good solution is to use polysaccharide, because, for example, cellulose, it's the most abundant polymer in the world. But the problem with polysaccharide that all of them are hydrophobic. So if we would like to use this polysaccharide, we have to make some modification first, some chemical in the surface, or to combine this polysaccharide with other hydrophobic polymer. In our group, we start first we test first polysaccharide chitosan as coating materials, because it's second abundant polymer, it's easy to prepare, but we use it as inner coater, and we use also wax at outer inner. This is because the chitosan, it's really hydrophobic, it absorbs water, and we cannot slow the release only by coating with this composite. For that reason, we use paraffin wax as outer coating, but even with double coating, we have total release of nutrient after 16 hours. It's not enough to tell that our coating agent it's for slow release. For that, we took to use our coating, our preparing PCL nanocomposite that we prepare in first, because we can control the hydrophobic character. So that's guaranteed the slow release. We have good mechanical properties. We will not have failures. It's easy to prepare a polymer. I guess that it's good markability of these coating materials. We can increase or decrease the filler contents to have this swelling behavior, and also our nanocomposite, as the filler and the polymer are biodegradable, is biodegradable. So we coated the granules only by deep coating, In THF, unfortunately, one drawback of our nanocomposite that the polymer is soluble in THF, and by deep coating, and as you can see, we have really thin films, and we have a good adhesion even with hydroxide nanotubes or with guar-gum between granule and the coating film. This is a good thing for us, and we studied the release study of nitrogen from uncoated and coated fertilizer in water, and as you can see, this is the red. This is uncoated fertilizer. We lost or almost all fertilizer after 1.5 hours. But as you can see, with 1% we have almost really good slow of nutrient. We need almost 50 hours to release the total nutrient instead of only 1.5 hours to release the nutrient from uncoated fertilizers. This is a good thing, but which we observed that when we increase the fillers, the release is a little bit faster than with neat polymer. This is maybe because of the hydrophilic character of fillers, or maybe because of low molecular weight of the polymer. Where we are making now some investigation on the effect of molecular weight against release, and also we are making some study on the crystallinity, if the crystallinity of the polymer affect the release or not, and we are making also some cross-linking to have more stable polymer for that. As a conclusion, during this study, we success to prepare cyclic ester in presence of the filler in open air. We get really high molecular weight. The same results show that we have a good dispersion of fillers inside of the polymer. We have a good dispersion, allow thermal stability and mechanical properties, and that granule will successfully coated by deep coating process with PCL guar-gum or PCL grafted hydroxide nanotubes. The liberation rate of nutrient could be controlled by adjusting the fillers contents guar-gum and hydroxide nanotubes, and this coating increased the nitrogen release in time to more than 50 hours instead of only 1.5 hours for uncoated granules. I would like to thank my collaborators, Professor Rehan, Mehdi Khouloud from OCP, Taha, my PhD student who is working in this field, and also Professor Bini Azzardwan from EMCSP for characterization of some materials, and also my financial support, OCP, CNRST, and Alexander Van Humboldt Foundation, and thank you very much for your attention.
Thank you, Professor Hsini, for this nice presentation. I think we are ahead a bit of time, and we can have one question from the audience. I guess we have one. From a cloakroom. Thank you, Professor Hsini. Very nice presentation. The use of these nanocomposites in agriculture is promising. Can we apply these products as foliar fertilization or as vector of certain fertilizers?
Good question. So no, not this kind of polymer, but we are developing some smart polymer, like thermosensitive polymer, in which it's changed the solubility in water independent of the temperature. For example, we have preparing one polymer. It can form micelle in water, for example, at 30 degrees, and it precipitates at high temperature. But with this polymer, this nanocomposite, it cannot be used for. We can use also oligoketosan as for it, mainly for nanoparticles, but that one we cannot use it for foliar fertilization.
Okay, I would like to thank you, Professor Hsini, again for the nice presentation. And also, it will be my pleasure now to really before leaving the stage to the closing remarks session, which will be just right after this session, I would like to thank the presenter again of this session entitled Sustainable Developments of Nanotechnology in Agriculture. I would like especially thank the organizers of the workshop, Professor Abdullah Karoum and also Professor Ardwan Miezza. And please stay with us for the closing remark, and thank you again.
Hello, and good morning, evening, or afternoon, depending on where you are in the world. It is my pleasure to welcome you to the closing ceremony of the Phosphate Days to 2020. My name is Yasser Biaz. I'm the General Director for UM6P Ventures. It's the VC arm for UM6P. So we are delighted again to receive you to conclude on this beautiful event. Before I go into the agenda, Just wanted to place this in context. So this is a biannual event that takes place in collaboration between ForestNet and UM6P, and this is the second edition thereby Phosphate Days 2020. And so this is a great opportunity for us to host the grand family and ecosystem around the phosphates industry and derivatives. So without further, before I relay you on to my colleagues, let me please just give you at least an overview of the agenda to come today. So I'm going to relay to my colleague, Dr. Qussair, who's been essentially the host for this whole event, so that he can provide his closing remarks. And then I'll discuss two particular topics on Young Researchers Days and the Talent Moonshot program before I relate to my colleague who will explore further with you the entrepreneurship and innovation programs that we have at the university. And I'll take again the chair at least of the presentation to present also what we do in terms of ventures and support for entrepreneurship programs, and then we'll announce the happy winners. So without further ado, please, Dr. Qussair, it's yours for the closing remarks.
Thank you, Mr. Chairman. Good afternoon, ladies and gentlemen, dear colleagues and friends. Mr. Chairman, distinguished guests and participants of the Phosphate Days 2020, it's a great pleasure for me to say a few words at the end of this conference. On behalf of the organizers, Mohammed VI Polytechnic University and ForestNet, it's indeed my pleasure to make a few closing remarks and express gratitude to all those who made this virtual event a reality. We would like to welcome you here at Mohammed VI Polytechnic University campus, but unfortunately the unusual condition of COVID-19 that we are experiencing at this moment have forced us to hold this conference in virtual form. We had thought of postponing it to a later date, but our commitment to organize Phosphate Days every 2 years led us to organize this event on the date we agreed upon during the previous edition. Organizing this conference in virtual form has been a challenge for us as organizing committee, but after 5 days of presentations, knowledge sharing, debate, and exchange, I can say that we have succeeded in meeting the challenge. The International Conference on Phosphates was organized around plenary lectures, keynotes, thematic sessions, workshops, and posters. I know that there have been many interesting and useful presentations these days, both in the plenary sessions and in the thematic sessions. According to the chairman and the rapporteurs, whom I thank warmly for their sincere involvement in the success of this event, many new good ideas and experiences have been shared, good lessons learned, and important recommendations for research and partnership arose. A conference becomes a success only if participants are present. They are the lifeblood of a conference. Thus, we do express our sincere thanks to all of you who have attended from over 20 countries. We do thank you for being interested in the International Conference on Phosphates. You have done an outstanding job sharing with all participants your valuable knowledge, insight, and experiences. Your presentations and active participation are the success of this event. I would like to thank the Mohammed VI Polytechnic University staff. Without their involvement and assistance, this conference would not have been possible to arrange. A special thank you goes to the Mohammed VI Polytechnic University Department of Communication, who played a key role in disseminating information about the event through national and international medias. I would like to express our deep gratitude to the scientific committee, They did a wonderful job by selecting the best communications with rigor and impartiality, which makes ICP a conference of high scientific value. I also want to express many thanks to PR Media, the agency that accompanied us during the preparation and holding of this event. Their contribution greatly contributed to the success of this event. Finally, I would like to thank each member of the organizing committee. They had the courage to keep the conference on the agreed date, and they were working hard to make it a success. A special thank to the UM6P TTO team, especially Fatim Zahra, for ensuring that Phosphate Days event ran smoothly. I would also like to emphasize that feedback from you is essential to evaluate and improve in the conference. I hope that we will be able to show real progress when we reconvene this conference in 2 years. We have learned that collaboration and solidarity is the best way forward to achieve challenging goals. I therefore hope we will all leave Phosphate Days event with renewed vigor, determination, and the enthusiasm to rise to the challenges before us. Thank you so much, and stay safe and secure.
Thank you very much, Dr. Qusayr. So I'll take it from there, and thank you very much. Of course, we cannot thank enough all the team that's done an immense kind of work to make this happen. So without further ado, I'll start shuffling the slides and going through the presentation. And so the Young Researchers Day, as many of you may know, or if you did not know, I mean, essentially the Phosphate Days is a combination of essentially two blended or aggregated events. One of them is the ICP, the International Conference on Phosphates, but together with it and adjacently, there's the Young Researchers Day. I take particular interest in this event because this is its second iteration, and I took very active part in the first one. And it's been something quite interesting. And so the whole idea in the Young Researchers Days is, adjacent, or a couple of days, it was earlier in the week, there was this event taking place. And so the whole idea behind it is to encourage, those aspiring PhDs to continue to augment, hone in their skills, but there was particular focus in this iteration on technology transfer, and so that's all the kudos to the TTO team that's been pushing for this dynamic approach. And so the whole idea was, again, to explore those technology transfer opportunities and match them with the industrial ecosystem. And so as part of the process of training the participants in the Young Researchers Day, there was a lot of work on the IP side of things and the technology transfer, process methodologies. There was also a lot of work in the communication aspect of scientific research, so its valorization. Also, developing or practicing some entrepreneurial soft skills and the like, but also looking into business models from maybe a couple of different angles. The talent moonshot. So forgive me, just to essentially, I should have said this. So the Young Researcher Day event sort of took place for the second time. It started with the first iteration of Phosphate Days. The Talent Moonshot is essentially a different program that sort of aims at identifying high potential PhDs and to get them more interested in postdoc opportunities and positions and within the UM6P ecosystem and grander. And so essentially we use the aftermath of the previous iteration of the Young Researchers Day to identify a grander pool of talent. And so we kept in touch with them, we brought them in, and so we did some work here with them and some particular training for them, with regards to, essentially projecting on their post thesis, developing, creativity angles, innovation, develop also better sense and sensibility to intellectual property. Valuation, of course, and valorization of research is paramount, of course, there. And so if you would allow me, I just have to look again at the agenda because I cannot cheat my way and look into it right now. All right, so yeah, with that doing, so I'm sorry, yeah, with that doing, so let me explain to you how the Young Researchers Program kind of works. So we started with 20 candidates, and of course the pool was much, much grander before that. And so the jury was composed of very well-rounded and balanced areas of expertise from the jury members, and it was as much in intellectual property, in entrepreneurship, and then more, scientific fields, some of them more vertical. The whole idea was to, explore the novelty, the innovative aspect or characteristic in the project. The project candidates, the person also, the PhD or the candidate that is defending their work. And so we look for, at the same time, acumen or skill in scientific excellence, but also in presentation skills. But also, number three, how feasible and practical, pragmatic the project is, and especially relative to the readiness level, let's say MAP. But also, and of course, finally, the financial aspect. So we thinly selected, some 6 candidates that we will have an opportunity to present to you a little bit later. And so at this point, I will relay to my colleague, Adnan Alawi-Suleimani, who is leading most of the programs, if not all, in entrepreneurship and innovation at the university. So you may have come across his name with Impulse, founders, and many of the other programs. And so it's yours, Adnan.
Ladies and gentlemen, good afternoon. I would like to start by thanking the organizers of the Phosphate Days 2020 for inviting me to this ceremony. So my name is Adnan Alawi-Suleimani. I'm in charge of startup incubation and acceleration programs within Mohammed VI Polytechnic University. As you probably already know, OTP Group, the company that sponsors our university, has played a significant role in the emergence and the development of the Moroccan entrepreneur system, and continues to do so today via its university. Our entrepreneurship and innovation platforms aim to unite and support a global community of students, researchers, and visionary entrepreneurs who are committed to the development of our continent. We have created an investment company that is run by my colleague, Yasser Diaz, UM6P Ventures, to support the projects of students and researchers. We have also developed a number of startup support programs. Impulse is one of them. Impulse is a program that has been developed with the MASH Challenge to support companies and startups working on African challenges in the fields of agri-tech, biotechnology, mining technology, and material science and nanotechnology. We have also launched Explorer, an incubation program in partnership with MIT Sandbox Innovation Fund to help develop startup projects within our campus. We are regularly organizing training courses for our students and researchers to help spread the entrepreneurial mindset within our university and to help valorize the research works that are carried out within our university. We also have created doctoral school in the fields of management, entrepreneurship, and innovation. And we have recently inaugurated an incubation space of 3,000 square meters to host and incubate startups belonging to our ecosystem. U Founders is part of the programs that are being developed within the entrepreneurship and innovation platform of the university. We deploy pre-incubation, incubation, and entrepreneurship around awareness programs for our students, researchers, and also for the entrepreneurs that belong to the university's ecosystem. The Youth Founders program supports both deep tech and shallow tech startups. The deep tech track is dedicated to startups providing technological solutions based on substantial scientific advances or high-tech engineering solutions. The shallow tech track is reserved to startups whose business models are based on known and already existing technologies. Digital startups are a prime example of the type of startups that we incubate within this track. The Youth Founders initiatives revolve around three main programs. So we have UCAN, UDisrupt, and UScale. UCAN is a program that aims to spread the entrepreneurial mindset within the university. So throughout the academic year, UCAN deploys a series of lectures and conferences with role models and successful entrepreneurs, as well as series of workshops that are led by experts on topics related to entrepreneurship and innovation. The UDisrupt program is a pre-incubation program that aims to test the maturity of the projects and to help the entrepreneurs to overcome the obstacles they may encounter during the early phases of their entrepreneurial journey. The ambition behind such program is to support entrepreneurs towards the realization of a marketable product with customers and initial traffic. And then we have UScale, which is an incubation program that aims to transform mature projects into companies. Such program is dedicated to project holders with confirmed potential. The UDisrupt and UScale programs last typically between 6 to 24 months and offer a series of theoretical and operational modules aimed at helping aspiring entrepreneurs develop successful companies. Training and mentoring occupy central position in the entrepreneurial support we offer to aspiring entrepreneurs and innovators. Our mentors are seasoned entrepreneurs and subject matter experts with a deep understanding of the matters related to the businesses of our incubated startups. We also offer our entrepreneurs access to a best-in-class co-working space within the campus of the university. Our entrepreneurs can also access the university's Fab Lab and living laboratories to develop new products and services. Youth Founders provides these entrepreneurs with the opportunity to pitch their solutions to potential clients and partners. It also assists them in their fundraising efforts by putting them in contact with the venture capitalists and business angels who might invest in their startups. We have launched the first cohorts of the program. July this year, we have 12 startups that are currently incubated within the program. 3 of them belong to the deep tech track, while the remaining 9 are shallow tech startups. So the 3 deep tech startups we are currently incubating are AgriSafe, BioChemir, and Phosphate. So AgriSafe is a startup developed by our colleague Mariam Rafiqi, who is a plant pathology researcher at Mohammed VI Polytechnic University. AgriSafe researches and develops tailor-made, safe, and affordable organic solutions for sustainable plant protection. AgriSafe helps farmers protect their crops while minimizing the use of pesticides. We have BioChemir, which is also among the startups that are currently incubated within Youth Founders. BioChemir develops DNA sequencing technologies and diagnostic kits that help with the screening of infectious diseases. We have Phosphate, which is part of the first cohort of the program. Phosphate produces silane materials from rocks obtained from phosphate mines, and these startups also helps reduce the environmental impact of phosphate mining activities. The 9 shallow tech startups of the program are AeroDrive, AgriEdge, Ecolit, Emior, DocHome, Ghibli, Marsetne, Nuafiknov, and Sones. So AeroDrive is a startup specialized in the manufacturing of drones that are used in defense, mining, precision agriculture, and geological exploration. AgriEdge provides recommendations to farmers on fertilization, irrigation, and treatment of plant diseases to help them increase their crops, increase the yields of their crops. Ecolit is a startup project by students from the coding school 1337. Ecolit develops a digital waste management solution for households, hotels, cafes, universities, etc. MER connects smallholder farmers to customers and ensures product traceability, and also provides an innovative supply chain model from collection to production to delivery. Doc Home is a telemedicine platform that connects patients with doctors and provides them with medical advice. The platform also facilitates the self-diagnosis and the monitoring of critical or suspected cases of COVID-19. We have also Ghibli, which is among the startups of the Youth Founders 2020 cohorts. Ghibli is a web and mobile platform where users can order homemade meals prepared by housewives who are passionate about cooking. We have Marsetne as well, which is an e-commerce website based in Mauritania that markets electronic and cosmetics products. We have New Afrique Nov, an entrepreneurial initiative from OCP that is developing disinfection device based on UV rays for objects that cannot be disinfected with chemicals. And we finally have Sounds, which is developing robots for lawn mowing, which saves times and cuts costs and improves the quality of lawn mowing. We'll be launching the call for applications for our second cohort soon. So if you have an idea, passion, or a technology that you would like to develop into an entrepreneurial project, then please reach out to the Youth Founders team by email or through our website, u-founders.ma. We'll be more than happy to support you in transforming your entrepreneurial ideas into reality. Thank you very much.
Thank you very much, Adnan. That was quite interesting. So in the same fold, then I'll continue presenting a little bit further and give you a little bit more context. So I apologize, just wanted to check that everything is fine you're seeing. Okay, so I mean, essentially the way at the university we want to look into innovation and entrepreneurship is that we do not want to look just at one component. It is our responsibility to make sure that we have a continuous stream from when an idea can come about all the way until it can access the capital's market. So in a way, the way we have kind of structured this is that at the university is essentially the haven for the innovation forces and the entrepreneurship forces. The innovation forces, and you're witnessing these. I mean, we have education in all forms and shapes and modes. And so in addition to that, I mean, of course, we have the labs and the living labs. So to stay within the context of, well, at least the theme of at least a Phosphate days. So we have, I mean, of course, the advanced labs, but we also have like an experimental mine. We have an experimental chemical plant, which is an operational environment. And say also, in energy and things like that, we have further infrastrucures where even, let's say, for the mining industry, can get access to more advanced platforms to continue to test out your hypotheses or, your innovation, opportunities or projects. And so marry that to forces of entrepreneurship. And we've had the good opportunity here to listen to Adnan present on Youth Founders, and he did, of course, present also an Impulse, but also Explore is one such program that is resident here. And this is a program that we developed with MIT. So in the logic of that continuous stream, please just again regard that we have innovation. And so all the infrastructures, the talent, and the projects that go there, entrepreneurships to seed those projects into more tangible, say, business opportunities. But then our responsibility, of course, is, as we said, to make sure that we have a capital outlet, and that's that. So just to, again, go one more time over this kind of innovation and entrepreneurship kind of forces. And so on the innovation side, if we were to consider that on the first column that I showed, you can see here that I really carry at heart, essentially the Young Researchers Program and Talent Moonshot. So we were inspired even to build this slide as per the work that was done on Young Researchers Day, or at least competition 2 years ago. I was lucky to get one of the 3 winners, laureates of that program. She's in my team, and she's the Science and Ops lead. And we took also another person who, another colleague hired internally. So talent is really important. We do cater to that. We have a lot of functions that relate to technology transfer and all that relates to IP. And so that's clerking, doing the quants and the qualitatives in terms of trying to understand, the stance of an IP, an opportunity and an IP, but also opening up routes to market. And that's really important. And of course, the Technology Transfer Office plays an immense role here. And the other thing which my colleague Adnan mentioned is that, essentially the university is this kind of aggregate environment where you have, everything that you would essentially need as ingredients for your entrepreneurial endeavors, infrastructures, the skill enhancements, the engagements, internally and with partners. There's, of course, events such as this one. We have access to data within the grander, let's say, data ecosystem of the university. We have Fab Lab. We have an advanced also innovations lab for operations that can also do a lot of prototyping. We can do that as well with Masir, with the Green Energy Park, talent, and the market. And so please just allow me here now to talk a little bit about about Explorer and where this comes from. So, essentially, MIT has this fund and a program that they aim at their undergraduates and graduate students within MIT. They've been doing this for 5 years, and we have historic relationship and philosophical affinity with MIT, and we decided to pursue this novel opportunity jointly, and so as to simplify even further maturation or graduation out of an entrepreneurship program and starting to access capital. And so Explore is 2 components. It is one program, and the lead for that is Adnan, my colleague, as part of the innovation and entrepreneurship platform within the university. Explorer with MIT and by MIT was designed originally for education purposes. And as I said, it was aimed straight at that layer of students. With MIT now, we're trying something different that we're testing at our university, or at least we have tested in the first cohort. Now the second cohort is initiating, and we plan to scale this at some point. The difference was in the verticality of the model is that we do not address just the undergrads and the grads. We address all of the student base completely. We do researchers and staff as well. So there's that verticality, but also horizontally we spread. Why? Because we're not just within the strict confines of UM6P. So it is the grander family of UM6P. And so we have Masir with us, which is the Foundation for Advanced Science and Research. We have the Green Energy Park also associated with it. We have the 1337/UO Code schooling system, which is a hacking school. So we've gone deep and wide with MIT on this one. And so the whole idea is essentially to leverage the same network of mentors, MIT side, but also on our side, cross-pollinate them. So cross-pollinate the mentors in the first phase, and then downstream at some point, we're going to be mixing up teams from MIT and from the partners that are associated with this program. And so what we do is that this program runs in cohorts. So it's 2 cohorts per year. First one started in first trimester, quarter of this year, 2020, just concluded like a month ago. And the new cohort, Adnan is launching the new cohort now. In the next couple of weeks, it's going to be probably closing. So we have multiple formats to engage with the students, or at least, I'm sorry, with the startuppers. And so the whole idea is to identify, so essentially we tier them as per level of maturity. And at that time, we'll match them with a Moroccan mentor and an MIT mentor. And so when I say those mentors are not strictly university personnel, for example, in the case of MIT, these are very remarkable people that are associated, volunteering their time to give a little bit also to these inspiring entrepreneurs. And so we're happy to be able to provide such a program. And so yeah, please just also reach out to the webpage of the university. And when you look into the innovation and entrepreneurship platform, you'll see this one and many. And so in the first cohort, just to say that one, in no time, we've been able to gather the interest of 262 people. You have all the analytics here that can show you at least the breakdown of, essentially who's been, applying. It's mostly students. I think that was kind of expected. And also, we're very happy to feature a lot of the 1337 new code, which is the hacker schools. And so that, of course, arranges a number of different topics and areas. And so, yes, of course, we were just like anybody else impacted with COVID, because the whole idea of this program is essentially to decomplexify access to capital in such a way that, capital may become accessible, as the project matures and it's been built. And so the more fundamental components of this program were essentially mixing up people, which we could not do with COVID, but also a lot of market exploration. So there was a lot of kind of, field outing type of activities, which we could not, satisfy. But at least one thing we're happy with is that we've been able to condense a lot more people gather. We've been able to cross-pollinate teams, and so match, teams few with others. And so I should not have shown that. And so, yeah, now, just before I go to announcing the winners, I would like to, again, encourage, please, everyone to really take advantage of the resources that are offered by the Technology Transfer Office. For addressing scientists and engineers, please do, consider that we have, very seasoned teams in everything that relates to innovation and entrepreneurship. Any which way, we will have a program for you. And so now, before I announce the winners, I just wanted to echo at very high level the efforts and the praise, for the efforts of all the teams that have made this possible. And so, without further ado, now I'm going to announce, in no particular order, the three people that or persons, projects that have made their way through this Young Researchers competition. So by no means is this just that there are only three winners, but at least the three individuals here have been able to demonstrate or at least impress the jury. And so please let me announce them at this point. And so I'll announce them in a first pass, and then I'll call them one by one so that they can make a 5-minute presentation. And so please join me in congratulating Ms. Safa Sadawi, Ms. Amina Amrai, and Mr. Tariq Labilta. And so please, Safa, will you take over and at least show us your work for 5 minutes?
Good afternoon, ladies and gentlemen. First of all, I would like to express my gratitude and happiness for the award received during this competition. So my name is Safa Sadawi, postdoctoral at Masir, Moroccan Foundation for Advanced Science. I participated with the project entitled Multi-Target Detection using 79 gigahertz radar for mining applications. The main purpose of this project is the digitalization of OCP mine by providing mining machines with anti-collision radars operating at 79 gigahertz. This is in order to improve the productivity and to increase the safety of operators and machines. Well, my main mission in this project was or is to develop high-performance detection algorithms that are operational and in the dust mining environment. Young Researcher Days 2020 was a very great experience for me. I'm delighted to have participated. It's reassuring for the future of R&D in our country. Such events encourage and motivated us to continue and excel. I thank the organizing committee for their hard work and devotion. Special mention goes to Mrs. Fatima Zahra Fouad and Mrs. Kautar Louzi. Thanks to the scientific committee for the deep revision of our works, and many thanks to the speakers who shared with us relevant instructions for the success of our project. Also to the judges for their remarks and the points of improvement to bring to our topics. Thanks a lot for all my colleagues in Master. You have all widely contributed to the success of this Young Research Days 2020 edition. Thank you.
Well, thank you very, very much, Safa. So at least knowing that you're working in those, high millimeter waves, at least you can reconvert to 5G if you don't find anything in the mining business, right? Anyway, so thank you very much. So now I will invite Amina Amarai to please join us now and speak your part.
Thank you, sir. Hello, everyone. Firstly, I would like to thank all the organizing and scientific committee, Fatima Zahra and Kautar Louzi, as well as the jury of the Young Research Days 2020, for giving me the opportunity to present a part of my research project, application of inorganic materials for the removal of heavy metals in aqueous medium and acidic medium. So this work is a part of AFOS project supervised by Professor Azzi and Professor Ashuri, which I would like to thank them for sincerely for their leadership, support, and efforts during this work. Some of my scientific research during my PhD focused on elaboration of inorganic nanomaterials by different techniques in order to purify different mediums, including phosphoric acid. So the additional problem treated during this project is contamination as aqueous medium by heavy metals such as lead and cadmium. The second medium is phosphoric acid, which also contaminates by certain heavy metals during manufacturing process. This major problem allowed us to research an efficiency solution. We avoid all the inconsistency present in some methods currently used. So this project focused on development of new inorganic nanomaterials based on sample non-toxic reagents characterized by strong absorption properties to remove heavy metals. So the treatment process provides a total decontamination in short processing time of treatment with the highest stability. So this research project will be destined essentially to phosphate industries and water treatment industries. This will provide economic and environmental impact, specifically in Morocco. So finally, during this excellent training, I will thank the organizing training. By the high-level trainers, we have developed in communication and valorization of our ideas on project. The training also encouraged us to research, to have the mindset to create our startup in future. For this opportunity, I will also thank all the team for the quality of the training, for this precious time. It was really a very good experience.
All right. Well, thank you very, very much for that. And please join me now in welcoming the last but not least, Tariq Labilta.
Thank you, sir. Good morning, good afternoon, and good evening, everybody, depending on your local time. My name is Tariq Labilta. I am a PhD candidate at Qadiyat University of Marrakesh, and I am glad today to share with you an innovative application of glasses, especially phosphate glasses, throughout our project entitled Elaboration, Characterization, and Application of New Phosphate Glass Fertilizers. As you know, yesterday we celebrated an important event, which is World Food Day. This is an opportunity to discuss the notion of food security and to answer one of the most questions, which is how to feed 9 billion people by 2050. Yes, it is expected that world population will attain to 9 billion people by 2050. And researchers believe that we should produce 70% more food to deal with the growing demand and production in all over the world, and especially in Africa. One of the options to reach this goal is by improving the fertilization strategies. Fertilizer industry is in a challenge to improve the efficiency of its products. This is done either by improving the fertilizers already in use, or by developing new type of fertilizers. And this is what our project is about. We developed new type of fertilizers, which are controlled release fertilizers, especially vitreous fertilizers. And let me show you some achievements that we have been able to achieve during this project. The first one is we have been able to elaborate glasses containing the maximum of nutrients necessary for plants. These glasses have been made at low temperatures and with cheaper raw materials to reduce their cost. We have managed to control the release rate of nutrients by modifying the chemical composition of glasses. Also, most of the elaborated glasses follow the standard of controlled release fertilizers. Application of these fertilizers have been made on some crops with great economic and social interests, like wheat and tomato in the first step, and they have significantly improved the growth, yield, and physiological parameters compared to conventional fertilizers. Finally, I would say that young researchers' days with the team to meet technology transfer challenges was one of the most best scientific experiences that I have ever attended, for two reasons. The first reason is because it was an opportunity for me to share, discuss, learn, and discover new researchers in phosphate domains, and also because it has changed totally my vision to my project. This is done through the discussion, questions, and feedback of the jury members, and also because it has because of the sessions and the trainings we had, and during which I have learned a lot about intellectual properties, about entrepreneurial mindset, about business model, etc. For that, I would like to thank the organizing committee and to congratulate them for their great work, for their dedication. I would also thank my team at Qataiyat University, and especially Mr. Mohammed Nsnawi. Thank you, Phosphate, please.
All right. Well, thank you very much. Thank you very much, Tarek. So now we want to announce the three winners of the Best e-posters, and so these winners have been elected by way of vote. So yeah, I will say their names. So you have Aisha Rudari, who's done some work, or at least in her work is related to agriculture and biotech. She's affiliated with UM6P, and I think I can just invite you to read some, at least, the affiliation. And so she did work on influence of salinity on phosphorus and potassium uptake of tomato plants grown in hydroponic conditions. The second position comes to Ayman Al-Mashi, on his work on valorization of phosphate mine waste rocks as aggregates for concrete. And so he is with the Faculty of Science and Technology, Qataiyat in Marrakesh. Third ranking goes to Ilyas Barrak on his work on synthesis of Moroccan Stevensite layer double hydroxide composite for heavy metals retention from aqueous medium. And he's associated with the Lab of Material Centrifugal Systems in Tetuan. And yeah, so yeah, please join me in congratulating the 3 teams. I think we also wanted to announce that the both 6 teams, so the 3 and the 3, will be able to benefit, or we're going to invite them to join some of our innovation and entrepreneurship programs. And so, Adnan, please, if you want to also have a more kind of explicit, announcement with regards to that, please feel free to do that. But the whole idea is, I think, for the previous, at least, the competition winners, the Young Researcher Day winners, are going to benefit or join this ongoing cohort of Explorer. And the 3 Bestie poster winners will be invited to any one of the other programs that we have. So we'll just check with Adnan if he has another word that he wanted to add to that, please.
Thank you, Yasser. I think that you've summarized it all. So the 3 winners are going to benefit from the Explorer program, which we are developing in partnership with MIT. And obviously, we are going also to welcome all the other participants to the other programs that we are developing within UM6P, so that we can help them turn their technologies, their ideas into reality. So we look forward to working with all of you on your entrepreneurial projects.
Thank you very much, Adnan. And so I guess we're getting now to the conclusion of our presentation. And so I wanted to have a word here to thank our sponsors, Arkelio and VW Bar, part of Aventor. And they are in the labs, equipment, and services. So please, we encourage you to go visit them as a way of thanking them for supporting this event. And so if we do not have any other comments or questions, please farewell. And thank you very, very much for having been with us here. Thank you again for the organizing team. As Dr. Hussein mentioned, without the audience, there is no event. And so we want to thank you for continuing to attend and participate with us. Congratulations to all the 20 candidates as well that participated through this. And so we'll continue talking to you, seeing you, and working with you. And Godspeed.